feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loo Lin School of Medicine Healthy Longevity webinar. Thank you for joining us. Today's guest of our show is Dr. Mitchell Lee. He will talk about how to identify healthy aging therapeutics. I think we all want them. Please use the Q&A function to send your questions and remarks. First, as always, we are starting with a short presentation, this time by Clayton Chu, an intern at the NUHS Center for Health and Longevity, and he will present on his project, really evaluating senescent cells and their phenotype and paracrine functions.
Thank you for the kind introduction. As mentioned, I'll be sharing my team's study on the characterization of the senescent phenotype in senescent cells and paracrine function analysis in the cardiovascular system. Singapore has an increasing elderly population, and 1/4 of Singapore's population is expected to be 65 and above by 2030. Combined with the fact that the prevalence of chronic diseases is on the rise amongst the elderly here in Singapore, it is becoming increasingly relevant to study the implications of aging in chronic diseases. According to the Ministry of Health, cardiovascular diseases are one of the leading causes of death here in Singapore, accounting for over 30% of deaths in Singapore from 2020 to 2022. We understand that aging is a significant and unavoidable risk factor for cardiovascular disease. However, the mechanism for why that is, and therefore solutions to this issue, have not been found. One of the hallmarks of aging is senescence. When cells continuously divide over a long period of time, they accumulate DNA damage and eventually senesce, no longer being viable in the healthy tissue. The secretory profile of senescent cells also changed, with the production of senescence-associated secretory phenotype, or the SASP secretome. Accumulation of senescent cells and its SASP factors, which is typical in the elderly, could lead to disruption in cellular processes and tissue function, resulting in a higher risk of disease. However, the SASP profile of senescent cells in the cardiovascular system has not been elucidated. Therefore, we are inducing senescence in cardiovascular cell types to better understand the aging process in the cardiovascular system on a molecular level. There are various senescence induction methods used in research, and we chose to utilize drug-induced senescence due to the practicality and reproducibility of the study. During the study, we induced senescence on cardiovascular cell types using a drug and verified it through senescence-associated beta-galactosidase staining, or SA-beta-gal staining. SA-beta-gal is a well-reported senescence biomarker regarded as a standard to detecting senescence. After we confirmed the senescence of the cells, we collected the cell secretome and its RNA. The secretome contains all of the secreted proteins released by the cells. The protein and gene profiles were evaluated using multiplexing technology, which allows us to analyze the levels of multiple gene and protein targets using only one sample. This saves time, samples, and costs needed to run the experiments. Proteomic evaluation of cell secretome was performed to analyze the changes in secretory protein expression between senescent and non-senescent cells. We analyzed the levels of 19 protein analytes, which are classified under two umbrella terms, inflammation and cell function. we found that there was an overall decrease in the secretory protein concentration in senescent cells. However, pro-inflammatory cytokines interleukins 6 and 8, which contribute to inflammation in senescent cells, were significantly more concentrated in the senescent cell secretome, suggesting higher amounts of inflammation which could have contributed to the loss of cell function. Transcriptomics evaluation was performed to analyze the differences in gene expression between senescent and non-senescent cells. We analyzed 18 gene targets and found that genes related to cellular function were mostly downregulated in senescent cells. An exception to this was the CDKN2A gene, which codes for the P16 protein and is normally reported to be overexpressed in senescent cells. In this case, it is observed through gene expression that the P16 gene is indeed significantly upregulated in senescent cells. What's next in our study? We are looking into the mechanisms of senescence propagation in the cardiovascular system. To elaborate, this will be done by investigating the influence of the SASP factors which are produced by senescent cells on healthy cardiovascular cells. The exposed cells' secretory protein profile and gene expression profile will be analyzed for any further changes. This will bring us one step closer to unlocking the molecular mechanisms of cardiovascular aging and designing interventions that mitigate cardiovascular disease in old age. I've come to the end of my presentation. Thank you for your attention. I would like to acknowledge Dr. Rufaiha, Dr. Marek, Ms. Sabrina, and my lab mates for their guidance and contribution to this project.
Thank you so much. Very nice presentation. Hopefully you will find the therapeutic. And we stay with the therapeutics. It's my honor to introduce to you Dr. Mitchell Lee. He is the Chief Executive Officer and Co-Founder of Ora Biomedical. And Dr. Lee identifies healthy aging therapeutics while building the connections between aging and diseases. And he investigates how genetic variations modify therapeutic efficacy. He earned awards for science communication, multiple NIH training grants, and the Gilliam Fellowship for Advanced Studies. And of course, you would like to know what Ora Biomedical is. It's a company that uses robotics and AI to perform high-throughput healthy longevity intervention discovery. And they are doing it in nematodes. This leads, hopefully, to novel interventions for humans and also for our dear pets. Thank you so much, Mitchell, to be here. I know it's very early for you. Thank you so much for logging in from Seattle and making the time to be here.
Excellent. Thank you, Andrea. Great to be here today. Great to be able to talk to you about what we're up to at Ora Biomedical. And thank you to the excellent science to get us started. Really interesting to think about, really how we engage biology from cells, understanding those insights, and where we come from at Ora Biomedical, thinking about how we can utilize, as Andrea mentioned, nematodes, or a small little roundworm, to get at fundamental mechanisms that drive aging and targets to modify those and small molecules that can modify aging. So before I get started, I want to thank everybody from the NUS webinar team, Andrea, Brian, Joe, Camille, Casey. Everybody has been excellent. They've been great digital hosts, and it's really excellent to be here so early, even though it's so early, I would say. So I have a little bit of a different kind of science talk for you today. I hope it's a fairly uncomplicated talk. At Ora Biomedical, we are drug hunters. We are seeking out the interventions that can be put into use, maximizing our healthy lifespan. The small molecule interventions and other kind of environmental dietary interventions we've identified to date really point at the potential of targeting aging to maximize our health, but we're far from realizing the full potential and promise of these types of therapeutics. So I don't think I have to belabor this point too much for this group, but there's a fundamental connection between the diseases that limit our lifespan and the fundamental mechanisms that drive aging. So these highly conserved molecular and cellular features that destabilize and drive aging ultimately lead to conditions like heart disease, cancer, neurodegeneration, and metabolic syndrome. So the idea in geroscience goes that by targeting that root cause, these molecular and cellular drivers of aging, we can also have positive impacts and delay or maybe completely get rid of the worries of these different diseases that limit our health. This isn't just the foundations of a biomedical revolution. It's really an important commercial proposition as well. So longevity medicine and longevity therapeutics is a growing industry. It unites multiple different sectors, including companion pet health. And really, it's been suggested that the longevity economy, as it emerges, is going to ultimately be one of the largest and most complex economies that has ever existed. So it's really a remarkable time to be doing these studies. And so where are we at? What are the problems? Why have we not, already found these interventions and put them into use? Well, there's a fundamental problem in terms of discovery science. It's difficult in this current funding climate for, really the last 20 years to get fundamental discovery science moving forward. And, I liken our progress in longevity interventions discovery to using hand tools, digging in the ground of chemical space with a shovel. And, that has allowed us to find a few different interventions. But what we really need is a large scale approach. We need the industrial backhoe to comb through chemical space as rapidly as possible, find these interventions, and then figure out how we move them forward. At Ora Biomedical, we think we've created the best backhoe, which we call the WormBot-AI. So this is a robotics platform combined with machine learning AI that performs live animal phenotype calling and tracking. So we look at and discover interventions in a live animal system, and we do it by focusing on what's important, what matters to us, lifespan and health throughout the lifespan. And we measure these directly. So this robotics platform and this initial work spins out from Matt Kaberlein's lab at the University of Washington, and Ora is has the exclusive commercial use rights for the platform. So it's about this point in the talk when people start getting curious about a fundamental question. What can we learn about human aging by studying something so seemingly different as a nematode, a small little round one? Well, actually, we can learn quite a lot. And so I want to give you just a little bit of an extended introduction to the nematode, or as it's been called in science, nature's gift to science. This is a small animal system. You can grow it rapidly in the lab. It eats an easy to culture food source. You can do cryopreservation in these animals. So you can freeze these animals indefinitely, pull out populations, and have live animals to study and work with. They've got a short lifespan, and the tools available to the nematode biologist are truly stunning. There are libraries of animals, each one with single genes deleted, non-essential genes. You can order those directly. You can order RNA interference compounds that will target and turn down gene expression. You can look at GFP tag proteins. Those animals exist and are easy to purchase and send to you. There are also a wealth of different disease models that accurately recapitulate human biology. When we think about aging and we think about ultimately applying it to humans, we're going to ultimately have to grapple with natural genetic variation. Humans are all different. They're also environmentally different. We have different diets and nutrient compositions. So this is going to be a complicated thing. There's a lot of noise that's going to make it difficult to translate these interventions. To find the interventions, we think it's best to compress that noise. And nematodes are actually perfect for this kind of system. Every nematode that you see in a dish is genetically identical and anatomically identical, all the way down to the very cells in the animals. So this gives us incredible signal-to-noise ratio to find interventions that impact aging, learn about those, and then start translating them forward into mammals and other animal systems. They're kind of referred to as a simple animal. I take exception to that. They have well-developed systems, including reproductive, nervous, muscular systems, and you can learn quite a lot about biology by studying these animals. As I've mentioned, aging is something that manifests first at the cellular and molecular level. It destabilizes evolutionarily conserved processes, and that's ultimately what drives tissue and organ level dysfunction. So when you go all the way down to the cell level, this is my question. Is this a cartoon of a human cell or a C. elegans cell? Well, it's a trick question. It's both. A C. elegans cell has all the same organelles as a mammal cell. It has the same fundamental problems. It has to take in nutrients, convert those into the cellular building blocks. That's an imperfect process. It generates waste, and that waste has to be dealt with. So at this fundamental level, this level where we also see destabilization and aging first beginning to manifest in molecular dysfunction, human cells and C. elegans cells are indistinguishable. Thinking about aging in particular, you know it when you see it, you know it in a mammal. You can see these differences very clearly between a young and an aged animal. Same story in C. elegans. A young animal is in that top picture, and an old animal, it just looks like an aged animal. Thinking about it through the lens of the hallmarks of aging, it's really not a surprise, or it shouldn't be a surprise, that so many of the hallmarks of aging are associated with aging in C. elegans, because we built the hallmarks based off of, in part, this animal system. So it's actually helped us understand and really provide fundamental insights into the basic biology of aging that we see translated into and working in mammalian systems. For Ora, we think the most important aspect of using C. elegans is the conservation of longevity interventions. So the gold standards to date in biology of aging are really dietary restriction and mTOR inhibition. These are things that work, not just as far back as animals and nematodes, they work as far back as fungus. So these highly evolutionarily conserved features and interventions that modify those features can be identified and studied using these systems. And my last point is really a pragmatic one. In terms of getting the job done, combing through chemical space, which is millions of molecules, this is really the animal system to do it in. It's fast, it's inexpensive. I think there's probably some strong ethical reasons not to raise millions and millions of mice to do fundamental drug discovery. And at the same time that you're doing these analyses in C. elegans, you can also look at health span and do parallel disease modeling as well. So they're an incredibly powerful system. So I've kind of told you the story, read you the riot act about the value of C. elegans. Now I want to turn my attention a little bit to what we've been up to at Ora Biomedical. So we've been doing our fundamental drug discovery operations for a little over a year. In terms of our internal development, we've already identified at least one molecule that is surpassing the current gold standards in terms of longevity interventions. So I'm not going to actually tell you too much about that. I'm going to tell you about something very different that we're up to, something that we call the Million Molecule Challenge. So this is something that we really started, this idea came about as we were moving the WormBot out of the Matt's lab into a private sector. And, at that time they were thinking, wow, we could do thousands of interventions with this. We could test tens of thousands of molecules potentially with the WormBot. Once we got our hands on it at Ora, we wanted to continue innovating around this platform, figuring out how to scale it. We were very successful in that regard. And so as we were doing our back of the envelope math, it started to become pretty clear that not only could we surpass tens of thousands of interventions, with the right resources and a very short period of time, we could test up to 1 million interventions. We can start doing that large-scale work of combing through chemical space. And, I say the right resources. So we're still generating those resources. There's opportunities to help play a fundamental role in helping us advance this science. So as we started thinking about the scale that we could reach with the WormBot platform, we wrote up a paper, we presented this to the scientific community as, in some ways a field-wide challenge, but we see at Ora Biomedical a pathway where we can directly lead the charge and complete the million molecule challenge ourselves. As we continued to think about it and get excited and really think about how to bring more people in, this idea came about that I'm really proud to be a part of. It's sponsoring the Million Molecule Challenge. So this is a really innovative grassroots citizen science approach to doing this kind of fundamental interventions discovery. So you can go to the Ora Biomedical website right now. You can follow that QR code. You can go and you can sponsor an interventions test for a small fee, $100 for a single intervention, $200 for a combination intervention. You can select among FDA compounds and other natural products, and you can put us to work. You be the PI, you be the scientist, we're your team of researchers. You pay this fee and we'll return to you a high quality data set showing how this intervention works. We'll also put it in an open access database. And so I also want to mention that we're proud to partner with Lifespan.io, who's our official charity partner of the Million Molecule Challenge. This is a fundamental thing that we're doing at Ora to advance drug discovery and accelerate it for everybody. And we're accepting donations to make that a reality. So just over the last couple of weeks, we've released our version 1 database of the Million Molecule Challenge sponsored database. And so you can explore that here today. I invite you to have a peek while I'm going through the rest of my talk, and you can see what we've been up to. So for the rest of the talk, I want to tell you just a couple of stories that have came out of the database so far. One of them, we had a lot of enthusiasm for people wanting to test rapamycin. Rapamycin, part of the gold standard right now for longevity interventions. We tested it, and we didn't see an effect. At least not at the dose tested. So this is something that we were kind of surprised, excited to see. And it wasn't just rapamycin. When we looked at similar chemistries, temsirolimus, everolimus, these rapalogs as they're called, we failed to resolve lifespan extension. Now, I say this, a little gleefully because I know what the punchline is, but it's fun to make these kind of fundamental connections between thinking about, what's been out there. So let's work through this. Let's take a step back. Okay, does this mean mTOR inhibition doesn't work in nematodes? No, it doesn't mean that. There's plenty of genetic evidence that shows that turning down mTOR signaling, particularly complex 1 signaling, extends nematode lifespan. These are a couple different genetic studies that show the case. So is it something about rapamycin? Is it something about that chemistry in particular? In fact, we think that's what it is. So rapamycin, you see the structure there on the slide. It's a pretty gangly molecule, and it has a pretty poor solubility profile. So what we wanted to do to make this case was do a spot assay. So this is one well. These are the environment that our animals live on. It's an agar media, and what we do for our drug dosing is we put the drug right on top of the media once it's already solidified and poured. So we add the drug topically and it soaks in. So we did this with rapamycin, and we started doing this dose response. You see this row here is 5 micromolar to 30 micromolar. You see at 5 and 10 micromolar, not much. It just looks like a pretty standard plate. At 20, you're starting to see speckles emerge on the media, and at 30, it gets even stronger still. When we did larger doses, 50 micromolar and up, we saw this very strong precipitate forming. And 50 and 100 micromolar is about where you see published reports of rapamycin extending lifespan. In our hands, this creates a condition where the animals are really swimming over crystals of rapamycin. It's actually impacting their ability to get to their food source. So there are studies, we're not sure if in those studies they found ways to solubilize it and make it more soluble. There's some other techniques you can use that might help the effective concentration. But in our hands, when we looked at 100 micromolar rapamycin, we didn't even test it because, one, the WormBot system, visualizing these animals through time, we just couldn't get a good visualization of the animals. And even if we did a manual assay, it's really difficult to decouple the influence of dietary restriction or mTOR inhibition on the lifespan extension that's being produced. So this is something that we're writing up. We're going to publish this, bring this out to the field, and just give us the opportunity to think about mTOR inhibitors in general. So does this mean that no pharmacological mTOR inhibitors work, or that they'll all precipitate out? No, it doesn't mean that either. So this is a different mTOR inhibitor that was sponsored. Thanks to Rapamycin Longevity Labs for sponsoring this. This GSK molecule, it's a dual kinase PI3K mTOR inhibitor, and at the same doses of rapamycin and the rapalogs, we resolved robust median lifespan extension. And so this really begs the question in our mind, are we using the best mTOR inhibitors for extending lifespan? That's something that we just don't know. There hasn't been a broad concerted effort to screen through every mTOR inhibitor. There's hundreds of them. And so really, I hope this is a humbling moment for us to think about where we're at in the field. This is the most promising target for extending healthy lifespan. And it's not clear that we've even found the best small molecule for that existing target. Happy to talk to you a little bit about why I think we've not came as far as we could have in terms of understanding even mTOR inhibitors and their function in healthy lifespan. And it's worth noting that we think that so far what we've seen is this is the first report of this GSK molecule extending lifespan, and we'll be publishing this up. So these are the top interventions to date. This GSK molecule leads the list with a pretty impressive 35% increase in lifespan. The two other tied for second compounds are two natural products. Berberin, which I think the Kennedy lab has done some work on, and this compound called punicologan. I apologize if I butchered the pronunciation. This is a compound that's found in pomegranates. So two different natural products also produce pretty robust longevity extension in the C. elegans system. And so kind of larger summaries of where we're at today. So the Million Molecule Challenge database is pretty far from 1 million molecules. We've got 72 interventions in it to date. We're going to be adding in combination interventions in the next couple of weeks. We're figuring out how we modify the database essentially to incorporate those right now. Of those, these are all community-sponsored, community-requested interventions. About 12.5% of those extended lifespan at the doses we tested in our model. And a couple of them significantly decreased lifespan. It's kind of interesting to think about crowdsourcing information and getting ideas of longevity tests, and how that might skew the percentage of lifespan extending interventions we identify. So it'll be interesting to compare this number to our unbiased internal screening that we're doing as well to see what this hit rate looks like and how it changes. So to conclude and start wrapping up, I hope I've convinced you that C. elegans is really the ideal model for where we're at, rapidly screening and finding longevity interventions. And if not, I hope I've given you the opportunity to think about the fundamental importance of this animal system really is forming the basis of a lot of our knowledge in the biology of aging. You can get involved with the Million Molecule Challenge. This is an exciting effort that we're underpinning and moving forward. And so far, we've already found an mTOR inhibitor that, at least at the doses that we've tested, extends lifespan much more robustly than even the gold standard. So next steps, continue screening. We want to continue testing, finding surprising interventions that extend lifespan, and thinking about how we combine multiple interventions together to really create remarkable increases in healthy longevity. We're building out of this the world's largest, most rigorous, and most experimentally consistent open access longevity interventions database. This is going to have not just longevity hits. This is going to have chemicals that show no effect, chemicals that decrease lifespan. And we are very excited to think about how predictive AI can be used with these incredibly high quality databases and data sets we're generating. And you can get involved, and we want you to get involved. So the team at Ora Biomedical, the strategic directors are myself, Matt Kaberlein, who's all this research initially spins out from. He's now the CEO and co-founder of OptiSpan. If you haven't looked up their YouTube channel, I highly encourage you to have a look. And John Saby, who's a visionary biotech investor and really has helped us move these studies forward. At the ground level, getting the work done is my partner in this, Ben Blue, who's our CTO and the architect behind the neural net architecture for identifying and phenotype calling in our WormBot-AI system. And the hands making the experiments happen are Director of Invertebrate Research, Michael Mears. We've got a great team of advisors as well. You'll know Brian Kennedy and Jan Gruber from NUS. They're co-founders of Ora Biomedical and help us think about our scientific direction. Jason Pitt is the inventor of the WormBot platform, and we've got many other investment partners, grant support, and we've been really fortunate to get involved in a lot of different commercial accelerators. We're members of various different groups, and it's been quite a process to go from being an academically trained scientist to an entrepreneur leading a longevity biotech company. And these groups have really helped make that transition possible. And so I want to thank you all for your time. And with that, I'll be happy to take any and all questions.
Thank you so much, Mitchell. Wonderful presentation. I think a wonderful setup to really speed up things in our field. So well done. May I ask you, as I could, I think, now donate and say, this is my molecule I want to test. And I think it's not increasing the health span of the nematodes, but it's decreasing them. So would you accept these kind of molecules to actually showcasing that these are toxic for our body?
Oh, absolutely. Yeah. Our system is going to help us resolve all manner of different small molecules. We can do biologics. You can turn down gene expression with RNAi very readily in the nematode system. And we're excited to work with academic collaborators. So we've got some collaborations with Dr. Alatin Kaya, who was on the webinar not too very long ago. And even with the public, if you are interested in something, you can reach out to us. Some of our constraints are we need to source it through a reputable chemical supplier and have it shipped directly to us just so we know exactly what we're getting. And then, yeah, we can work together and do the experiment.
But you're also focusing on the ones who are not deemed to extend the health span.
Absolutely. Yeah, no, I think, thinking about health span and lifespan, if we find interventions that increase lifespan but don't show a corresponding increase in health span, those are interesting. If we just look at interventions and find those that don't increase lifespan or even have a negative impact, those are interesting as well for a couple reasons. They inform our AI. I think that is going to help us create a better data set to allow us to have more robust prediction. The second thing that's important is, nematodes, they're helpful in understanding the biology of aging. A very different feature of nematodes is there are parasitic nematodes that are actually a human health concern. If we find novel ways of treating parasitic nematodes, that's also a benefit, and that's a positive health impact. It makes the story of longevity biotech and what we're doing at Ora a little more complicated to tell. So we don't lean on that as much, but we're still excited to find it.
Yeah. So imagine after this presentation, there are thousands of requests coming in. How do you select what comes first?
So we haven't had that problem yet. I'm excited to have that problem. Our system is very, very scalable. And even right now, the rate limiting step isn't getting an experiment on the WormBot. It's getting the hands to set up the experiment. And so as we scale that up, there's, we're going to see what kind of team we'd need to grow to actually even feed all the WormBots we have in our V1 fleet. But yeah, if we get to that point where we have a lot of enthusiasm, we're going to be excited to scale, bring on more people who can help us out. Undergraduate research was a big thing that I focused on as a PhD student. I ran teams of 10 to 12 volunteer researchers, and I would love nothing more to grow a similar kind of team at Ora.
Let me ask you a little bit a critical question. Reproducibility sometimes is a problem of the interventions being tested in animal models, from one lab to the others. And of course, there can be lots of different explanations of it. Temperature, how you feed, what kind of model you use, etc. How do you take care of that within your company? So how do you test if it's reproducible, even in your own system?
Yes, absolutely. So we do think that we're doing things in a way that's going to consistently and reproducibly generate good, reliable data, but that's in one set of hands under one set of experimental conditions. Aging is very complex. And I think a part of that, why we see this differences is part of the underlying biology. And there's some things that we're not tracking that if we tracked them, we might be able to control for them a little bit better. That's one of my intuitions. But what we do at Ora is we do a lot of replication. We do replication studies, we do dose response studies, and we're putting out all of that data. In the open access database, we're going to make that available, and we've got videos of it as well. So, when we do in fact see this variation, or maybe an intervention that does work once and then just completely doesn't work again, we can go back to the videos and we can look at those and really give ourselves the understanding of the ground level truth of what's happening, and then we can move forward from there. So I think there is a lot of unexplored biology that this seeming irreproducibility is pointing at.
Okay, so every experiment will be out there
for
without
the
any selection
database as part of the Million Molecule Challenge. We also run our internal discovery and development, which, we also want to make that available to the community as well. We just have to build in the step of putting in some IP protections before we release that out. So, once we can, make a claim around, aging and a compound, then we can release it and really, bring it out to the community. So we want to accelerate longevity interventions as a public good, while at the same time being able to move our commercial operations forward, because that's going to be at the heart of us getting these interventions, into mammal systems, into the trials that are necessary. We need the resources, so we need to protect some of that IP so we can leverage that to fund ourselves going forward.
You already mentioned dose response. Very often, if I see the field, we are testing a molecule with sometimes 2 different dosages. How do you actually tackle that problem? So we have different dosages, but also we have the frequency, how often we give it, when we give it. And especially if you're talking about mTOR, that's very important, but also the duration of the treatment. So what is your strategy to really nail it, what the biology is, and therewith when to intervene, how to intervene, and which doses it's and which frequencies, and when to start, for example?
Yeah.
Let's pick your brain.
Yeah, no, that's an excellent question. And the system is amenable to those more complicated dosing strategies. Right now, what we're doing is adding the drug onto the media and then putting day 1 adults onto that media. So they experience the drug from early life going forward. And that's another thing that we don't talk about as much with rapamycin in particular, is how short-lived it is. So within 36 to 48 hours, it's very likely that most of the rapamycin is not active in that system. We haven't been able to show that as clearly in the nematode system, but it's very clear in the yeast system where I did a lot of my fundamental PhD work. You can just see how the rapid degrades, and you can measure yeast outgrowth as a measure of that. So what we're doing right now is early life going forward. We can do late life things as we have an intervention. For the dosing side of the strategy, what we aim to do is really dose the animals at the highest concentration we can to resolve an effect, and then start going forward on a dose response from there. Nematodes have a pretty thick cuticle, so it can actually be a little difficult to drug them. So we hit them pretty hard. And we do a situation where we look at the solubility profiles of the drugs. We get the maximum predicted solubility in an aqueous solution, and then we guide our initial dosages around there. So typically what you'll see is about 10 to 100 micromolar, which is pretty standard across what you see in nematode literature.
Let's talk a little bit about this very tiny worm, C. elegans, how good it really is. One of your, I think, pros of using it was the large reproductive capacity. If I'm looking at humans, I think 20 years max. So, and most of women don't do that anymore, which is also good. But tell me, how does that relate? So is it really a pro?
Yes. So in terms of it being a model system that can be easily and quickly reared in a lab environment, that's a great benefit. In terms of recapitulating human biology, definitely. Well, so I'd put it this way. In terms of reproductive stress, the size of an egg to an adult nematode is very similar to the size of a 9-month-old baby to a human. So, the nematodes lay 2 to 250 eggs. So, the reproductive stress is massive. What we do is we actually turn down reproduction in this system. So what we do is we have them on a compound that prevents egg laying. There's also genetic mutants that you can use that turn down egg laying. Some people argue that that is building in artificial changes into this natural system and really changing it. We think that that's acceptable for us because we think that it is driving it closer towards human biology. We're getting rid of one of these major causes of death for the animals and helping us reveal underlying aging mechanisms beyond just this massive reproductive stress that they undergo.
So maybe before going to Casey, I see lots of questions from the audience coming in. My last question, I would love to see the translation from C. elegans to other animal models if necessary, but hugely into the human space. How much life expansion in C. elegans would you like to see to actually put in your money or your thoughts to actually translate it to other animal models? Is 7% really enough if you look at the complexity of C. elegans in the body and the real huge complexity we have as human bodies and human beings living in different environments, etc.? So where would you actually then say, okay, this is significant enough to transfer that molecule into the next model system?
Absolutely. No, our system is designed around that. So as you're looking through the database, you may notice that, we test about 50 to 75 animals per treatment. That's kind of a low number for an initial, not for an initial pass necessarily, but, if you look at the literature, a lot of times studies have multiple hundreds of animals that are tested in a survival curve. The benefit of us doing a low number of animals is it really keeps us focused on large effect size differences. So we have statistical power to pretty confidently resolve about a 15% and greater extension of lifespan. And we're really going after 15, 20, 25% and greater lifespan extending single interventions as our first pass. We think that there are large effect ones still out there to be found, and we want to move those forward. That's the low-hanging fruit. As we move forward, these small effect size interventions could actually be great for combination studies. And we're still thinking about how we utilize those, because that opens us up to a lot more exploration space to do. But it definitely hasn't escaped our attention that there are some other strategies that even these kind of more moderate extenders could be used for.
Thank you so much for that answer. I think it's so important not to throw out the ones only having a little bit of life expansion, because they could be so additive. I see lots of questions. Casey, our postdoc, please come to the floor and present the questions.
So thank you, Dr. Lee, for your insightful talk. So we have some questions from members of the audience. So the first question will be, how do rapamycin and calcium alpha ketoglutarate compare for you?
In our system, so with rapamycin, we weren't able to resolve a lifespan extension with that particular class of mTOR inhibitor, although other mTOR inhibitors did extend lifespan. I didn't show you the alpha ketoglutarate data, but we have tested that. We've resolved lifespan extension with it. It's a little bit more modest. If I'm remembering that one right, it might be somewhere in the maybe around like 10% lifespan extension that we did in that study. What we do is we start to catalog a lot of these longevity and lifespan extending interventions, and then we test our new large effect interventions with those combinations as a first pass to try to get at new additive and synergistic interactions. And alpha ketoglutarate is on our list.
Great, thank you. So the next question will be, the concentrations of rapamycin used are extremely high in micromole range compared to what is sufficient to see effects in mammalian cells, which is in nanomole ranges. Is it a physiologic difference? Is there a physiological difference between nematodes and mammals, or is the target effect that is driving this choice?
That's a great question. So, I think about accessibility of the compound to the molecular mechanism. Cells in a dish, you add rapamycin onto there, they've got a pretty expedited pathway to get into the cell, to get to the mTOR complexes. With C. elegans, as I mentioned, they've got a very thick cuticle. And so drugging the animals, you actually you go pretty high on the dosages. So that was an astute observation. Yeah, nanomolar ranges is what you see for yeast biology and all the way in cells in a dish. With animals like C. elegans, you're in this micromolar territory. We think that that's really a unique feature of C. elegans and just some of the challenges with dosing. So, we wouldn't move forward a study into mammal systems based off of C. elegans dosage. We have to do those studies in a mammalian system to start getting at the concentrations we'd want to test there.
So the next question will be, there are many factors that affect the C. elegans lifespan, like live or dead bacteria. The use of FUDR and the concentration of worms, how do you determine the standard experiment parameters?
Yeah, there's not really a literature that you can go to and say, this is the standard. Here's the way to do this study. You really have to engage the literature, and you've got to read deep in the methods, and you got to hope that the group was pretty detailed in their methods. For us, we do it the same way. We do FUDR. We need to turn down reproduction in this system because we don't transfer animals. So if you don't turn down reproduction, you get swamped out with progeny. And as Andrea and I were talking, we actually think there's a biological benefit to doing that in terms of making a model that more accurately recapitulates human biology. So that's one aspect. We've done different temperature changes. So in the database, what you'll see is some of the experiments were done at 25 degrees, and the control and the extension, they're a lot shorter lived. We saw that as a benefit early on because it allowed us to kind of expedite and go through screening quicker. It shortens the length of time for an experiment. We've started to move towards 20 degrees centigrade, which extends the lifespan of the animals. We're starting to think that might be a better background for looking at lifespan extension because, at higher temperatures, it's not clear if you're extending lifespan because you're improving fundamental aging mechanisms or because you're alleviating this heat stress that they're under. So the 20 degrees is what we've started to move towards. And our density that we use is always around 25 to 30 animals is what we shoot for. And we try to be as experimentally consistent with that as possible.
Great. So another question that our members are interested in is, what is the vehicle control used? Is it DMSO? Solubility of rapamycin are highly dependent on DMSO concentration. How do you standardize the vehicle control?
Yes. Most of the drugs that we work with are in DMSO. We aim to do a maximum of 1% DMSO for our single intervention studies. For the combination studies, you see that we do 2% DMSO. The stocks that we receive are already dissolved in DMSO, so that's kind of our way of getting the lowest dose of solvent possible. That DMSO extends lifespan also. So we think there's something about increasing permeability that makes the animals a little more druggable, and that we're seeing an effect on lifespan because of that. When you think about this going with rapamycin in particular, it's really informative to think about how we think about solubility. So in DMSO, rapamycin has a certain kind of solubility. You're adding DMSO into an aqueous solution. And so you have to really think about not just the DMSO solubility, you've got to think about the water solubility and think about the kind of weird chemical environment that you're putting these compounds into when you add them right on top of the media like that. With rapamycin, as you saw in the pictures, it precipitates out very readily. Other things absorb into the media pretty well. But that's a compound by compound thing, and something that we use a predictive solubility calculator to help us inform our dosing decisions.
Thanks. This is an interesting question. So would you advise that people, based on your results, are already taking supplements or drugs? How much caution do we need?
This is not medical advice. Do not take anything based on nematode lifespan. Be really careful about taking any supplements right now. I'm in my early 40s. I feel like I have the benefit of being very conservative right now in terms of how I think about my drugs and dosing and things like that. I don't take anything. There is nothing that I've seen right now that rises to the level where I would be very confident to take it. Rapamycin is close. And, once I start getting a little bit of targeted inflammation here and there, maybe a joint starts getting a little tight, maybe I'll start thinking about rapamycin there. But I personally, no. And I don't recommend people taking individual supplements necessarily. I really strongly advise against people taking combinations of supplements. I've talked to some people where, when I get into the conversation with them, they're like, let me tell you about my stack. And they just list off a grab bag of small molecules, natural products, and extracts that they take. And I have to tell them I don't know if any single one of those is beneficial for you, but I can almost guarantee taking all those hand over fist like that is not good for you. So I recommend the greatest level of caution. We're not there yet. We need the resources to get there. We can get there. And if you want to see it happen, we need funding to get there.
Okay. So should senolytic be tested in C. elegans?
That's a really interesting one. So in terms of senescence, C. elegans is a post-mitotic organism. What that means is they don't undergo active cell division throughout their adult life in their tissues. There's not from my understanding, and I'm not a senescence biologist whatsoever, it's not clear that there's a strong cell state that looks like senescence in C. elegans. On the other side, though, there is a secretory phenotype that has cell non-autonomous effects. And so I think aspects of senescence can be modeled in C. elegans, namely things like the secretory phenotype. but I wouldn't go so far as to say that a senolytic would have the same kind of efficacy that it might have with mammalian cells or mammalian tissues, which do have stem cells, are actively undergoing not just cell division, but also the senescence process.
We're also interested in what is the strategy to test the combination of molecules?
Yeah, so what we do is we do initial testing at one dose. We try to aim high to resolve a phenotype. If we see a phenotype, we move forward in a replication study, make sure it happens again and again. Then we do a dose response. We find the best optimized dosages, and then we start moving forward and then testing that compound against our panel of known lifespan extending interventions. So far, what we've seen with that strategy, and we're going to be releasing this data with some of the early sponsored experiments, we haven't resolved lifespan extension so far. A lot of them are with rapamycin, which is very interesting to individuals, but we didn't see the same effect with lifespan extension. We have resolved one that pronounced a lifespan shortening effect. So it's interesting to me to start thinking about how we understand combination space, not just to find these additive and synergistic interactions, but find the things that actually sum together to actually have a negative impact. And we're still figuring that out. And that's going to be that's one of the most exciting things that we're working on from my perspective.
Okay, thank you so much for answering the questions from our audience. So now I hand you back to Andrea.
Good. Thank you so much. This was exciting. I have one more last question for you. You have such an interesting career, really moving fields, and now being an entrepreneur. What kind of advice would you give to youngsters who are not yet in the field, who want to enter the field? What was your learning? What's your advice?
Yeah, the moment that kind of clicked for me was starting to have informational interviews. So I was a pretty dyed-in-the-wool academic throughout my scientific training. As I reached my postdoc, COVID occurred. I had an opportunity to kind of think about where my priorities were and what I wanted, and also the science that I wanted to do. I want to do this broad discovery science that, you get told you're just going on a fishing expedition if you try to write a grant around it. I want to go fishing. I want to find fish. So, figuring out how to do that. I started talking to some of the longevity biotech companies that are out there today. I had a really informative conversation with an individual over at BioAge who put the bug in my ear and got me thinking. They're like, at this point in your career, a lot of people are spinning up companies. And that just sounded so fanciful to me at that point in time to nearly be frustrating to hear it. It's like, well, great, I'll spin up a company, maybe I'll get a Pegasus farm, I'll start growing unicorns. And so I went and told that joke to Matt, who's been a long-term mentor and champion for me. And he said, well, the WormBot technology is about ready for prime time, and we're looking for somebody to lead it up. And it was just that right time, right place, right mentality that brought me to that opportunity. So start learning about the space. Go and talk to people in longevity biotech. Reach out to me. I make my information pretty widely available. Send me an email. Let's have a conversation. And I can talk to you about my pathway in more detail, and also how, your pathway might lead you into this space.
Okay, that's, I think, a great message. So listeners, please reach out to Michael Lee. Really great. Next time I will ask you your learnings about starting such a startup. I think there are also downsides. But yes, thank you so much for sharing your learning. Thank you for listening to this great webinar. Please leave any comments you have or suggestions, whatever you want regarding the show in the chat box. Thank you, Casey, for really sharing very nicely the questions. If you would like to participate in one of our studies, for example, our ORCID study, investigating the oral microbiome, yes, it's the oral microbiome, please be in touch with us. More details can be found in the end credits. Your participation is, as always, highly appreciated. We will close this webinar with a short video. And I can only summarize it. Being a father. Take care.
But my wife and I were in a new chapter of our lives. Got these two kids, 5 and 3. Got a 5-year-old and a 3-year-old, and I'm 49. I'm 49. There's an age difference of 46 years between my son and me, okay? Now, I love to play with him, don't get me wrong, but a lot of the play is laying down, all right? There's a lot of let's bury daddy tonight. His knees hurt, something's wrong with his shoulder. It's a lot of lay and play, right?
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.